Assalamu alaikum. My name is Maya Dehimi from International Islamic University Malaysia, College of Engineering. Today I'm going to present my topic about genotypic and phenotypic characterization of isolated marine bacteria and its potential to produce alkaline protease. First of all, we will start with an introduction, followed by the study background and the methodology used to obtain the results, which we will discuss, concluding with a small summary of the study findings. Marine environment is the greatest aquatic ecosystem and the most significant source of biodiversity on the planet. The biomass is much higher in marine ecosystems than in tropical rainforest. For instance, new technologies have demonstrated that there are around 20,000 species per liter of seawater, with 178,000 species falling in their 34 phyla. Hence, the primary widely available organisms in the ocean are microorganisms. Bacterial distribution reflects changes in the salinity of water, temperature, and other physico-chemical parameters. In the course of the bacterial diversity research that contributes to understanding Malaysian water, which is a part of the southern South China Sea field. This study is conducted of the Kuantan coastal area. So, South China Sea is adjacent to the Pacific Ocean and covers 1.4 million square miles. The sea is semi-closed area and extends from the Singapore Strait to the Taiwan Strait. This body of water holds 1/3 of the entire world's marine biodiversity. Much of these microorganisms are used in the variety of large biotechnological applications to provide new companies with innovative bioactive compounds and biocatalysts. What are proteases? They are a group of enzymes that catalyze the breakdown of proteins into peptides or amino acids. They differ widely not only in their functions but also in properties based on which they can be classified into various groups. Based on their catalytic mechanism, they can be classified into serine protease, metalloprotease, cysteine protease, or aspartate protease. Or based on their optimal pH, they can be classified to alkaline protease, basic or acidic protease, and also based on their site of action. There are three sources of proteases from animals, such as pepsin, trypsin, produced by the stomach, or chromatoid trypsin, plasmine, thrombin. From plants, example from papain or papaya, or also figs. And also the third source, which is microorganisms, we have the extracellular enzymes and the intracellular enzymes or proteases. Among the available sources for proteases, microbial proteases are currently receiving more attention to the technological and economic reasons. For instance, they are fast growing, sample culturing for large scale production, metabolically vigorous to secrete a large amount of protein directly into the fermentation medium, and also low cost media can provide micro and macro nutrients. So we go for the methodology. Starting with sample collection, so the seawater and sediment samples were collected in stirred containers from 4 different stations in the South China Sea, located at Kuantan, Pahang in Malaysia. Second is the bacteria isolation. So the seawater samples have been concentrated by filtration. On the other hand, 10 grams of marine sediments was added to 20 ml of stirred distillated water, vortexed, and after that the samples were incubated at 37 degrees Celsius for 30 minutes using an orbital shaker, and then centrifuged for 20 minutes at 5000 rpm. The proteolytic activities of all strains were assayed using the skim milk agar plates, then incubated for 24 hours at 37 degrees Celsius while observing the enzyme activity. So the positive colonies degraded the casein in the milk. Those positive colonies were sent to first base products and services for full-length 16S RNA sequencing. Phenotypic characterization of proteolytic strains were carried also out by observing the morphological colony characteristics and gram staining. And after that, a chemical characterization were conducted using a again microplates, which is microplates. So the isolates was grown on agar medium and then suspended in a special gelin inoculating fluid at the recommended cell density. The suspension is then inoculated into the GAN microplates, 100 micro ml per well, and the microplate was incubated to allow the phenotypic fingerprint to form. After that, the quantitative protease production, so 1% of inoculum was transferred in 250 ml conical flasks containing 15 ml of the enzyme production medium and incubated at 37 degrees Celsius for 3 days. Afterward, the supernatant was gathered and used as a crude enzyme after centrifugation. The results. So the first table represent the conditions of the 4 different stations physico-chemical parameters, which is set at different distances. So the station 1 at 400, 700, 1,500, so 1.5 kilometers and 2 kilometers from the shore. The temperature was ranging from 29 to 30 to 36, and salinity was from 36 to 37, and pH was around 8. So we have here a significant, we can see there is that there is a significant association between salinity and conductivity for all stations. It's primarily caused by variation in temperature, dissolved oxygen, and conductivity. There was also a connection between temperature and salinity. So when the DO, which is the dissolved oxygen, increase, we have the temperature and the salinity decrease. It was except for the station 1. So here we have 18 isolates from were isolated from 8 different samples, 4 samples from each seawater and sediments, so 4 stations. However, only 10 strains were found to produce. So only 10 strains were found to produce the protease enzyme by forming a transparent hydrolytic zone on skim milk agar. For the proteolytic strain were identified employing the 16R RNA sequencing, and this is the agarose gel electrophoresis for the 10 isolates producing proteases with their names. Also here we can see the chromatograph for Bacillus cereus. It's one of the identified bacteria and also phylogenetic tree. So we have all the chromatograph, the phylogenetic for the 10 bacteria, but this is just for instance. So the sequences of 7 isolates were reported in the GenBank database and their accession number shown in this table, and also the names of identified isolated and the isolated sources. So we have 3 from water, and the other 7 were from sediments. Then it has been matched with those available in NSBI database. So this figure represents the blast results against NSBA 16S ribosomal rRNA sequences. It was only for bacteria in the database. Here we have for Bacillus cereus 15. The first one is for the Bacillus cereus MD 152 and Bacillus licheniformis TB 212. The provided pictures serve for phenotypic characterization and colony description for the 4 different identified bacteria species. So we have 4 Bacillus cereus, 4 Bacillus lichenoformis, and 1 Bacillus staphylococcus and 1 Staphylococcus warneri. So as you see, these figures represent the biochemical tests results for on 94 substrates, 71 carbon source from the column 1 to 9, and 23 chemical sensitivity assays column from 10 to 12. All necessary nutrients and biochemicals are pre-filled and dried into the 96 walls of the microplate. All of the wells start out colorless like that. When inoculating, during the incubation, there is increased respiration in the walls where cell can utilize a carbon source or grow. So the negative walls remain colorless, as does the negative control in A1. There is also positive control well A10. This is this one. Okay, so the forming of purple a purple color represents a positive reaction due to the reduction of the tetrazolium redox dye. We have the broad line indicate that is a positive but not strong reaction. The crossed well here, the crossed well determines a negative reaction while it's supposed to be positive according to the ID database. For quantitative proteases, so this figure show the quantitative protease production results for B series. You can see that B series MD152 had the highest protease activity with more than 33 unit per ml, followed by the Bacillus lichenoformis IBN13 with 29 unit per ml. So both were isolated from station 1. In comparison, with no more than 13.5 for isolated from station 2, the lowest output was reported by lichenoformis TB212. So lastly, the conclusion, we can summarize that there was a negligible difference in the physical-chemical parameters of seawater between the 4 stations of the South China Sea, which have shown that the sea is well blended. 10 strains out of 18 isolates were able to produce protease enzyme. The isolates were identified as Bacillus cereus, Bacillus staphylococcus, Bacillus lichenoformis, and Staphylococcus warneri, based on the molecular identification on the 16S rRNA gene and biologen 3 microplate. So last, isolates of the MD152 and ABN13, Bacillus cereus and Bacillus lichenoformis respectively, were found to be the most promising proteolytic producers based on their quantitative enzyme production results. Thank you for watching. Assalamu alaikum wa rahmatullahi wa barakatuh.